Okay, so we should get going. Welcome back after our extended Easter Anzac Day break. Those who've been following their unit outline very closely, I'm sure you're all consulting that with great detail or not, will realise this was supposed to happen last lecture. That's fine. I have no intention of rushing through stuff just to get content through, so I'm more than happy to take a bit longer to go through the lectures we have, so that we're not rushing through. So that's fine, and it actually suits us better to start virology this week rather than start it late last week. What we'll end up doing is there'll be probably one case study, fungal case study we'll cut, but you'll get the full content that we need to get through for the course. Just to start to get everyone thinking and get your brain juices working, let's start off with a question. So you're all fired up and remember all your stuff from those earlier lectures. Who can tell me what's the difference or the definitions of, particularly the difference between a pathogen and a parasite, or pathogen, parasite, and a symbiont? So you've got these microbes that are associated with the plants. They can be called a pathogen, they can be called a parasite, they can be called a symbiont. Some of them may actually be called a pathogen and a parasite. But what is the actual definition? So who can give me a definition? What's a pathogen? Comes from the Greek pathos, meaning to suffer. Yeah, something that causes suffering to the plant, something that causes disease. So an organism that induces disease is a pathogen. So plant pathology, we talk about pathogens all the time. So these are the bugs that actually cause disease. What about a parasite? And what's the subtle difference? What does a parasite do? You know what
It keeps the host alive to feed off it, generally.
Doesn't necessarily keep the host alive, but it feeds off it. So an organism that feeds off another organism. So it's parasitic. So you can have in a plant pathology context, most pathogens are parasites, because most pathogens, they cause disease to gain nutrition for themselves. Can anyone think of an example? We had one in the example prac class of a pathogen that's not a parasite. There's only one I can think of. Do you remember that sooty mould example we had? So you have this fungus that's growing on the outside of the leaves of plants. You often see them often on lemon trees, for instance. They're actually feeding off exudates from insect pests. So the insect's the pest, the insect is the parasite, but they're feeding off all the goop that the insect's pooing out. So it's not actually directly feeding or parasitizing the plant, but it covers the leaves in this black fungal mycelia, blocks photosynthesis, and actually can cause yielding, can actually significantly affect the growth of the plant by reducing photosynthesis. So that is an example of a pathogen, something causing suffering, that's actually not a parasite. Saying that, pretty well all the other pathogens are also parasites, because that's why they do it. They're actually using the plant as a food source, and then doing so they cause disease. Can you think of a parasite that's not a pathogen? So something that derives food from the plant, but is not causing suffering. That comes down to our next criteria, which is the symbiont. So those symbionts, if you think about those, your rhizobia and roots, they infect the plant, they derive nutrition from the plant, but they also fix nitrogen and actually improve the plant's performance. So they're not pathogenic, but they are parasitic. Mycorrhizal fungi are parasitic, but they're not pathogenic because they actually provide benefit to the plant. There are potentially very minor pathogens that actually cause almost negligible impact on the plant itself, but kind of invade, some of these cryptic viruses, for instance, that can invade plants. They replicate in the plants. They're obviously deriving benefit from the plant, but they really cause a negligible effect, so you can't even measure it. So you could argue that they're not pathogens, but they are parasitic. Anyway, just to get us thinking. Let's start now with viruses. Viruses are fun. I like viruses. They are really, really interesting organisms. Here's a blatant advertisement. Okay, it's my book, so you can't get it in the bookshop here. They don't stock it for some reason. But if you want to get one, you have to go to the website at Cabby. But I like it because I wrote it. But anyone who's actually doing plant pathology next year, we will refer to it fairly extensively. Anyway. Plant virology. Why do we care? In terms of the numbers of plant pathogens, those that cause suffering in plants, probably the most diverse group of biotic organisms are the fungi, but they fall into a number of different groups. We'll talk about them later. The next biggest group will be the viruses. But also, importantly here is that some of these viruses, in fact, if you look at all of the major agricultural crops, in the top 2 or 3 diseases that affect them, one of them will no doubt be a viral pathogen. So viruses infect pretty well. You'll find virus diseases of all crops, and they're often some of the most important. We will talk about viroids as well and why they differ from viruses, but there are also some important viroid diseases. Plant virology itself as a science is not that old. We've known viruses for a long, long time. The first recorded symptoms of a virus was back in, I think it was an ancient texts from Japan in early, couple of hundred AD, or no, I think it was even BC, where they'd drawn this picture of this plant with these mosaics on it. It was clearly virus infected, and actually wrote a poem about it. And so that was sort of like the earliest recorded record of a plant virus, but no one knew what they were. The actual knowledge of what a virus is only about 100 years old, so that's actually not a long time really in terms of science. And really it's the breakthroughs in plant virology have occurred probably in the last 20-25 years, where the techniques for in molecular biology and the ability to isolate and sequence nucleic acids have got to the stage where they can actually start to look at genomes. And these guys have really risen to the forefront because these have got a very small genome, they're easy to characterise, and that has been very important in terms of understanding not only how viruses work, but also how their hosts work. But anyway, the discipline's about 100 years old. That's when the viruses were first actually discovered, seen, noted what the heck they were. Here's a cute little example. Everyone sort of call them Rembrandt tulips. All these old Dutch masters painting these still lives, and they have tulips in there, and they're all stripy, and they all look really beautiful. They're all virus infected. They're infected with a virus called tulip breaking virus. When tulips were first introduced into Holland, they're not a Dutch crop. They actually came from the Middle East. But tulips were introduced into Holland in the 1500s. And with their introduction, a few stripy ones appeared. So some of these ones that look like this, so they normally should be red, but these ones are stripy, and they're really pretty. They look beautiful. And those who had stripy ones were able to sell them for much more money than those that had pure red ones. And these became incredibly highly priced. So they're actually worth ridiculous amounts of money. They were selling them for things like houses, and they were swapping them for oxen. And the actual the actual value of individual bulbs became ridiculously large. It actually was one of the first futures markets, so people were actually selling the bulbs before they harvested them, and selling them multiple times. So they were creating an economic bubble, which we all know in today's world what happens with economic bubbles. You get the prices of individual commodities going like this, and everyone's making lots of money, lots of money, lots of money, lots of money, and then bang, it crashes and the world ends, and people dive out of tall buildings because they've lost all their money. Well, they didn't have tall buildings back in the 1500s, but they did lose all their money. So it kind of roughly coincided with this scientist, this Dutch scientist Clusius, who was showing that, hey guys, this stripy business is actually contagious. It's not a genetic variant. It's not this weird variety. It's actually something you can transfer. And he kind of cut a core through an infected bulb and put that core into a healthy bulb, and all of a sudden both bulbs were producing stripy flowers. And he's going, oh, so we can make these. We don't have to kind of just grow them up, because they're actually, tulips are really hard to grow. You know, you plant one tulip, if you're a really, really good tulip grower, you'll get 2 bulbs. If you're not a good tulip bulb grower, you might get 1 bulb, and if you're a really bad one, you don't get any back. So really hard to multiply. So if you only got 1 or 2 of these stripy bulbs, it's going to take you an awful lot of time to produce enough to sell, unless it's contagious, in which case you might be able to produce them a lot quicker. And of course, it's also spread by aphids, and so once they actually started to realise that you plant 1 or 2 stripy ones alongside a whole bunch of healthy ones, and then the next year when you harvest them they're all stripy, the value starts to go down. So in the 1600s we had the inevitable crash. The Dutch economy was destroyed, thanks to a virus primarily. But it wasn't until the 1920s they finally worked out what the thing actually was, and they could actually see for the first time this virus. Anyway, why are they important? The biggest factor with plant viruses is you can't cure them. Once you're infected, the plant's infected. It's not, you don't have fungicides, you don't have bactericides, you don't have nematicides, you can't chemically treat them. That's not completely true, but it's mostly true. You can't cure a plant from a virus in a crop situation. So once it's infected, it's infected. And so really it's all about prevention. And once you see it in the field, you're starting to write off your yield or write off the crop completely. So this is why it's actually becomes quite important, because if you discover it in the field, there's not too much you can do about it at that point. You've got to actually stop it getting there in the first place. Whereas if you've got a fungal disease and maybe you've got a few leaf spots, it might be possible that you can then put on a prophylactic fungicide and maybe treat it and recover the crop and actually get a yield. You can't do that with viruses. So a lot of effort goes around prevention and trying to avoid it in the first place. And that actually, if you go back to our first lecture when we were talking about cost, that's actually where a major part of the cost in relation to virus diseases occurs, is in this prevention, trying to avoid it in the first place rather than the losses after you've got the infection subsequently. It is hard to estimate losses, and if we just go down to yield losses and ignore all those other costs, you can see there's a whole bunch of examples where lots of things cost lots of money. Apple mosaic, I mean, if you've got an apple tree at home, it's probably got apple mosaic virus in it because it's very common, and a lot of even the commercial orchards would have it. But they can have significant yield reductions. Go down some other ones. Where are we? Here's a viroid, kadang viroid, which I think means death. Death. Either that or it's the sound of a coconut falling off a dead tree. But in the Philippines, effectively wiped out the coconut industry. 30 million trees dead by a tiny little piece of RNA. It's quite amazing that this thing can actually kill a tree. But as you can see, there's a lot of direct losses, and then you add into that all these indirect losses in relation to control and management. But the other thing that's fascinating about viruses is that scientifically they're really, really fascinating. These guys are at the threshold of life. By definition, and a lot of people's definitions at least, suggest viruses are not living things. They're not cellular. They don't have a cell membrane. They don't have a metabolism of their own. They are a piece of nucleic acid surrounded by a bit of protein. That's it. They are not. They have no ribosomes. They have no metabolism of their own. So they are very, very simple, but quite intriguing how they interact with the hosts. They make great epidemiological models because you've got infected or not infected. You don't have this, I've got a leaf spot, I've got something sitting on the leaf. Is that actually going to cause an infection or not? I'm not sure. You've got an infection or not. You can look at spread patterns with time and with space and actually do some really nice, interesting prediction models. They're actually a very simple organism, but very complex in the way they interact with their host. I liken, a lot of fungi, particularly bacteria, a lot of fungi too, are more thugs when they come to their interaction with plants. A bacteria will sit back and it will throw toxins at the plant cell, or throw enzymes at the plant cell. The plant cell will go and die, and then it feeds on the stuff that's left behind. It's pretty crude. It's let's stick a chemical and kill you, and then I'll eat what's left over. That's a pretty crude interaction. These viruses are intracellular parasites, so they get inside the cell, they hijack the plant cell metabolism, they change the way the plant expresses its own genes, they change the way the plant runs its own metabolism to make it into a virus factory for the virus. And so that interaction is incredibly complex, much more complex than some of these other pathogens. The other thing which is kind of neat is that because they've got this relatively small genome, you're talking about, plant virus is typically about 6,000 to 8,000 nucleotides long. That's not a lot. That's enough for about 5 or 6 genes only. And if we talk viroids, they're only a couple of hundred nucleotides long. They don't have any genes. They're too small to actually have a gene in them. So these are very, very compact, simple genomes, but they can do some amazing interactions with the plant. But because of that, particularly in the early days, people have been able to look at that entire genome and actually start to study, well, what does that gene actually do? Let's have a play around with that one. Let's have a play around with that one. Let's see what it does. And they've learned a lot about the virus, but they've also learned a lot about the host, because the virus interacts intimately with the host plant. And so when you change one of the virus genes, you see what it does to the host, and you can actually start to understand some of the host factors. So for instance, the way plants move molecules between cells was discovered through plant virology, because viruses need to move between cells. So they had no idea how plants do it until they actually studied this process. Anyway, what is a virus? We talked about them, but what are they? They're not that, but that's kind of what you get if you Google image what a virus is. I love this definition. It's completely erroneous, but I love it. The actual term virus dates back to Roman times, so well before they knew what they were, and they was defined as a poison, venom, ramish smell of armpits. That's great. Whitish, greenish, yellowish, all at the same time, which oozes out of ulcers, and it's not nice. It's horrible. That's clearly bacterial. It's not a virus. But that's when the term was first made. So virus was something that's a bit nasty and horrible and causes disease. Yuck. So these days, what really is a plant virus? Well, some of the characteristics of plant viruses. Firstly, they are obligate cellular parasites. So by that I mean they can't replicate outside of a cell. They must replicate within a host cell. They are cellular parasites, so they've got to be within the cell. They're not external to the cell. A lot of bacteria, we'll talk about later, live between cells. They don't get inside cells. A lot of fungi can penetrate cells, but also live outside of cells. These guys live inside the host cells. They replicate inside the host cells. They are non-cellular. They do not have a cell membrane. They are very, very, very small, sub-microscopic. You can't see them with a light microscope. So the microscopes we're using, you won't see a virus. You need to use an electron microscope to actually see a plant virus. They have a very small genome, and it's either DNA. Most of them are actually RNA genomes rather than DNA genomes, but they're either DNA or RNA, but they don't have both. So we've got DNA and RNA inside us. We've got a DNA genome, but we also produce RNA, messenger RNA to read our genes and all that sort of stuff. Viruses do not. They have a RNA or a DNA genome, but not both. They have no metabolism, so they can't make their own proteins. They need proteins, but they can't make them themselves. They steal, they hijack, they use the plant's metabolism. So the plant's ribosomes, the plant's transfer RNA, the plant's protein manufacturing machinery to make the virus proteins for it. And another aspect, which really is a nature of the way these are, the fact that it's non-cellular, they replicate from a pool of components, not by binary fission. What I mean by that is pretty well all cellular organisms, you start off with one cell and it divides and produces two cells, either two separate bacteria or maybe two cells that are still joined together. That's called binary fission, where a cell divides divides its genome, splits itself in half, becomes two cells. Viruses, they're non-cellular, so what they do is they produce lots of bits of nucleic acid, their genome, they produce lots of coat proteins, and then they stick the two together. So they coat the protein around the genome, and then there you go, you've got a new virus. So it's replication from a pool of components rather than by splitting a cell. I mentioned viroids. What is a viroid as opposed to a virus? A viroid, kind of already suggested, it's very, very, very small. So our viruses are small, these guys are smaller still. These are the smallest known biotic agent capable of inducing disease. Now I know in animal systems we talk about prions, which are just proteins, which are capable of inducing disease. These guys, I think, should be smaller than that. You don't get prions in plants, but they're certainly the smallest thing capable of inducing disease in plants. I don't think you get viroids in people either, so this is a plant thing. They are autonomously replicating. In other words, unlike, this is just a small piece of RNA. So unlike, say, a plasmid in a bacteria, that plasmid is an extracellular piece of nucleic acid that sits inside the cell, but it needs the cell and the enzymes in the cell to actually replicate it. It can't organise its own replication. This thing is a true parasite, a true pathogen, in that it can organise its own replication. So it infects a cell, it says, right cell, make a few million copies of me, and do it now because I'm taking over this cell and I'm the boss. So it is a true pathogen, it's not a hang around. They are very, very small. They vary from 250 to under 400 nucleotides. That is too small for a gene. They do not encode any proteins. They can't. They're too small. And obviously they're non-cellular. What do they look like? So here's some electron micrographs. So with an electron microscope, blown up bigger because it's on the screen, but it's probably, photo was probably taken about 100,000 times magnification. You can split your viruses into 4 roughly major groups based on their architecture. You've got these ones which are called rigid rods. So you look down the microscope and you see these sort of sticks, and they're all straight. Occasionally that's probably a broken one, where it's broken, or sometimes it might be a virus that's actually got a long and a short, you know, it might have multiple parts and a long and a short one. But there's a range of different virus genera that have these rigid rods. The tobaviruses, the tobamos, audios, etc. They're rigid. Then you've got a bunch that are flexuous rods. So again, they're stick-like, except these guys bend. These can bend. The other ones can't bend. If they tried to bend, they'd break. These guys bend. They will still break if you hit them hard enough. And they vary in length from shortish to much longer. And you've got, again, a whole bunch of different different genre. This is possibly equally the most abundant. This one's very, very common, particularly the polyviruses. There's an awful lot of polyviruses infecting plants. Isometric is the other very common virus type. These guys are little balls, or in reality, little icosahedra. Now, the number of faces on those icosahedra does vary with the virus type. Don't need to worry about that. You've got smaller viruses with less faces, larger viruses with more faces, but essentially they are ball-like. And again, a whole bunch of different genera that have this spherical or icosahedral type structures. Just out of interest, you'll see dark ones and light ones. Anyone have a thought of what the dark ones, why the dark ones are dark? Oh, I should say, so electron microscopy, if you remember, you put your sample on, your virus part sample, and then you flood it with an electron-dense stain, so something like phosphotungstic acid or something. And that kind of sits around here you are, and it sort of sits around here, the edge of the thing. So when the electrons come through, the image you get looks like that. So you've got this white bit in the middle, which is the virus, and you've got this dark stain around the outside, so you silhouette the virus. Anyone have a thought of why we might have some dark ones? These guys are actually hollow. So those guys, they're kind of protein balls packed full of nucleic acid. So that's a living virus, if viruses are alive. That one there is a defective one, so it didn't get any RNA in it. So it's actually empty. So you just got this protein shell with nothing inside. So the dye, instead of just sticking around the outside, has also gone inside and flooded it too. So again, it shows in the replication process a few slip-ups have happened. So that's the 3 main ones, and then the 4th category is everything else that doesn't fit neatly into one of the other categories, so what we call complex. And you've got a range of different types of structures. So here we've got ones that look a bit like a pill or a bullet, bacilliform. So they're kind of like an icosahedra at that end, and icosahedra at that end, and then the stretchy bit in the middle. So you've got these bullet-shaped ones. These guys here are round, but they're actually multi-coated. So instead of just being a simple icosahedra, they've got multiple coats around them, which seems very inefficient, given viruses don't have the capacity to make many proteins. So why use 3 or 4 proteins for coating yourself? I don't know. Ask the evolutionary background behind the virus as to why it decided to do that. But some of them have these rather elaborate coats. So like collimos, the cauliflower mosaic virus, for instance, is there. That one's alfalfa mosaic virus, that guy. These guys are cute. The Gemini viruses, or Bergamoviruses, as we tend to call them now, they're actually like 2 icosahedra stuck together. So Gemini, the twins, it actually looks like a little twin, and they've got these cute looking particles really. These are very, very important diseases, particularly in the subtropics and tropics, because they're mostly spread by whiteflies. And some of the most important virus diseases, particularly recently, are of that type. These guys are not quite flexuous rods. They're close, but they're incredibly long, twisty, very simple structures. The attenuoviruses. These last two, that's the tospoviruses and the rhabdoviruses, are interesting because these groups are both include species that infect plants. But also infect. I should take that back. These viruses invade cross orders. These are plant viruses, but they're also animal viruses. So these are pretty special in that they have the capacity to invade and replicate in both plant systems and animal systems. So in this instance, the tospoviruses, tomato spotted wilt is its type member. It invades about 1,000 different plant species. It's carried between plants by thrips, and invades the thrips and infects them as well. So it's an insect virus and a plant virus. Its family, the Bunyaviridae, pretty well everything outside the tospovirus, everything else is a vertebrate pathogen, including things that cause hemorrhagic fevers and nasty, horrible diseases in humans. So they're actually closely related to a whole bunch of animal and human viruses. Similarly, the rhabdos. These guys invade plants, but they also are carried by things like aphids and invade the aphids that carry them. Who can think of, on the name rhabdovirus, can anyone think of the type virus for this group, which isn't a plant virus, obviously? Probably a bit hard. Rabies. So rabies is actually a rhabdo, and obviously it's not a plant virus, but it is also one that invades both the insect that carries it and the host it gets into. So we've got these plant ones that invade plants, and evolutionary, there's some discussion as to where these came from. So you've got this one genre that invades plants, and all the rest of them invade animals, yet they also have the capacity to invade the insects that carry them. So is this an insect virus that's adapted itself to invade plants, or was it actually a plant virus that's adapted itself to invade the insects that carry it? Good question. And the reason why they're called complex is because they have these lipoprotein coats around them. To be able to invade animal cells, they need to infect them by a budding system where they come across, they meet up with the cell membrane of the animal cell, and they bud into it. So they actually need a lipoprotein membrane. So these guys all have a coat, and then they have a membrane outside of that, which they steal from the plant they infect. So we said viruses don't have membranes. These guys kind of do, but it's not a cell membrane. Viroids are small. So this is the entire genome. If you got really close here, you can see that's a UTA. That's the actual nucleotide sequence of potato spindle tuber viroid. 250 odd nucleotides. The striking thing about it, they are circular molecules, but they have a lot of this internal complementarity. So the sequence here is obviously a mirror image of the sequence there. And so it actually binds to itself and makes a pseudo double-stranded structure. So it's a circular molecule that kind of binds to itself internally. This is how they imagine it kind of looks like inside. Why this is really important is that this structure is incredibly tough. Now RNA is a labile molecule. So in a cell, RNA would normally be chomped up pretty quickly by the enzymes, the RNases inside the cell that are designed to chew it up. Because like when we make RNA, we've got a DNA genome, we make messenger RNA to express a cell. We want to get rid of that messenger RNA after we've made that protein, and so it gets chopped up and recycled. Viruses with a single-stranded RNA genome will similarly get chopped up, but they coat themselves in a coat protein to protect themselves. These guys don't have a coat protein. They protect themselves by making themselves pseudo-double-stranded, which is harder for the enzymes to attack, and then all this twisted. So the enzymes really can't attack that. There's no loose ends to grab, and they can't digest it. So these guys are very, very tough, and that's how they can survive without any protein protection. There's two major groups of viroids. There's the something or other. I've forgotten the name of it. Sorry. That have this more linear structure, and then you've got the avocado. That's terrible. Sunblotch, avocado sunblotch type, which is another group. So there's effectively two major groups of viroids. One of them has this a bit more complex structure where you still have that internal complementarity, but it's less linear and you've got more stems and loops and weird shapes. And the other one's much more structured. So two major groups of viroids that are present around. All invade plants, all cause nasty things. So that's what they look like. What are they made of? Here's a great quote from these guys. It's a Nobel Prize winner. Piece of bad news wrapped up in proteins. So effectively, that's what they are. They're pretty simple. They are protein surrounding nucleic acid. So the genome surrounding by protein. So let's start off with the outside bit, the protein. Mostly because it's a bit if you're looking at the electron microscope, that's the bit you see is the protein. It's a capsid or a coat protein, which is produced from a virus gene. So the virus has a gene which encodes in its genome, which will, once it infects a plant, makes the plant produce this protein. This is the protein that will coat around the outside of the virus. So here's a schematic of a rigid rod, and you see each of these little purple lumps. That's a protein. It's exactly the same protein. So this is a coat protein, and there's thousands of them that coat around to make the whole virus. Here's a schematic of an isometric one. You need a lot less proteins, but that one, all these yellow ones are, in fact all these ones here too, are all the same protein. So again, you use the one protein, it binds together to form either a ball or a linear structure to protect the nucleic acid. It has roles in protecting the nucleic acid, as I said. So that RNA molecule is sitting inside here and it's protected, so it can't be digested in the cell system. So it's important to make sure the virus survives and doesn't get chewed up by enzymes. It's also important for spread, for dissemination, for movement from plant to plant to plant, because this is the external part of the virus. So for it to leave, if all it can do is invade one plant and that's all it can do, then it's not a particularly effective pathogen and it won't survive. As soon as that plant dies, it dies. So it's got to get from that plant to the next plant, and it does so by a variety of means, and we'll talk about that in a minute. But the way it gets recognised and picked up is by the external part, this external coat protein recognising it's usually an insect vector that can actually recognise it, pick it up and carry it from plant to plant. So it has a role in both protecting the genome and in moving it from plant to plant. Most have only one coat protein because they can only make 5 or 6 proteins, so why waste your genome making more than one? But there are some that have more. Cowpea mosaic has 2. Clover wound tumour virus has 7 coat proteins. And this is a particularly big virus, and it needs more proteins because it's an icosahedra structure, and one protein type wouldn't actually make the ball. To actually physically make the ball, you actually needed 2 different, or in this case 7 different actually protein structures to actually make the ball shape. But it's a bit overkill. Vast majority just use one. From diagnostic perspective, the protein structure and the way it binds to nucleic acid is obviously important for us to tell which virus we're dealing with. One, it's external profile, so if we look down the electron microscope, we can see if it's a rod or a ball. 2, because it's also the bit that's going to induce the immune response if we're going to use something like a serological test. So if you're using serology like ELISA or one of those lateral flow tests, it's detecting the protein, it's not detecting the nucleic acid. We mentioned this before, the TOSPOs and the rhabdos are a little bit more complex. They have, so here's your RNA, here's your coat protein around that, and then they steal from the plant a lipid membrane type structure which will coat the whole thing, and then they put some virus proteins around that as well, and then the fully intact virus is now able to invade the insect as well as the plant. The important bit, of course, is the nucleic acid. So, and again, I believe the only plant pathologist who's ever won a Nobel Prize for plant pathology is a guy by the name of Stanley from the US, and that was for virology, for actually identifying the actual nature of a virus, except he got it wrong. So he purified this tobacco mosaic virus, and he showed that it was protein, that it's made of protein, and that's what it is, and he got his Nobel Prize. He missed the fact that it's actually got nucleic acid in it as well, which of course is the really important bit. There was a couple of others, including an Australian actually at the same time, who was doing similar work and actually noted not only the protein but also the nucleic acid component. So they got it right, but they didn't get the Nobel Prize and $1 million that comes with it, unfortunately. But if you look at the genome, the nucleic acid, by far the majority of viruses, plant viruses, have a single-stranded RNA genome. So a linear, single-stranded piece of RNA. 90% of all viruses are in that manner. Usually as a single molecule, sometimes they split it into multiple molecules. But in biology, there's always exceptions, so as soon as you say all viruses are like this, you'll find new ones. So yes, there are some double-stranded RNA viruses, there are some single-stranded DNA viruses, and there are some double-stranded DNA viruses. So we've got the full gamut there of the different structures, but these guys are by far the most common. They encode for virus-related functions, and they have multiple genes. So again, this is a schematic of the genome of tobacco mosaic virus, and in here you've got 1, 2, 3, 4, 5 genes. And the other thing you'll notice is that it's really efficient with the way it uses its genome. So it's not long. How big is that? I can't think, I haven't got it written up there. It's probably like about 6,000 nucleotides long. But instead of having a gene, and then a gap, and then another gene, and then a gap, and then another gene, or maybe even with eukaryotes where we've got introns and exons, and we've got all sorts of other structures. Here, you've got one gene with a read through there to produce another gene. You've actually got another gene here which sits inside, the coding of that sits inside that one. This one's overlapping with that one and that one. So you've actually got a lot of information packed into a very small space. And that's very typical of plant viruses. They have small genomes, but they pack as much into it as they possibly can. Taxonomy. I hate taxonomy, but anyway, we need to understand taxonomy of things so we can diagnose them properly. With viruses, we split them up into the type of nucleic acid they've got. Are they RNA or are they DNA? Do they have rods, flexuous rods, isometric particles, something different? Do they have one particle? Do they have two particles to make up the whole virus, so it split its genome into 2 bits? Are they enveloped or not enveloped? And more recently, because nowadays it's becoming much more important and easier to do, we can look at the entire genome. We can actually sequence the entire genome, and then we look at the way the genes are arranged. So a bit like here, we can look at, okay, you've got the polymerase here, you've got the coat protein here, you've got a movement protein here. That structure makes it look like a tobamovirus. If we shift some of these around, then it might be a completely different virus.
You just mentioned that the nucleic acid can be broken up in multiple parts.
Yep.
What kind of problems does that cause for replication?
You're going to have all of the parts in a single cell for the virus to work. So if you've got, like alfalfa mosaic virus has got 4 components. Ding, 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 ding. For that to infect a plant, you need all 4 in a single cell, and then it'll have all the genes it needs to actually replicate. If you only get 3, it won't work. So for the virus, it's actually a potential problem if it can't get all of them in. In reality, it works relatively simply because an aphid will happily carry, it'll pick it up and it'll carry enough particles, and you'll get more than enough into a cell. Why does it do that? Because it wants to get as much genetic information into as small a space as possible. Why viruses need to be small is because why plant viruses need to be small, not animal viruses. Animal viruses can be much bigger, much bigger. Plant viruses, here's a plant cell, need to get from this cell to that cell and so on. There's a dirty big cell wall in the way. So unlike in animal systems, where a virus can bud through and can actually move through the membrane and then move through the membrane to the next cell, in plant systems they've got to get through a cell wall, which is big, thick, like a brick wall. How are you going to get through that? Well, they get through the same way all molecules in plants get between cells, and that's little passageways between cells called plasmidesmata. So these plasmidesmata are cytoplasmic bridges that go between cells. They're pretty skinny. So an intact virus is too big to fit through. So they have to use, they have to actually modify the plasmidesmata, they have to actually split themselves up and squeeze themselves through. But if you want to get lots of genetic information, by packaging it into 4 different bits and then putting all 4 through rather than one huge bit, kind of makes sense if you're trying to be a big genome through a small gap.
So is it one of the reasons for plant virus being impossible to be killed?
The main reason it's impossible to kill is that once it's inside the cell, there's no obvious biochemical target because it's using the cell biochemistry. So yes, you can kill the virus by killing the plant. There are some chemicals which will stop replication, but that obviously stops the plant replication too. So you can actually stop everything for a while, and then hopefully, and in some circumstances, at least in a few cells, you can make a few cells virus free because the cell might start to divide without absence of a replicating virus. But an intact plant, you can't kill it because the only biochemical targets are the plant's biochemistry. So a herbicide's a great antivirus treatment. It's not going to save your crop though. Why do we care about taxonomy? Because I hate it, but why do we care? Because you need to know your enemy. If you don't understand what you're dealing with, you can't devise an appropriate strategy. So if you understand the group it belongs to, even if it's a brand new virus, if it's a flexuous rod that looks like a potty virus, you've by even just knowing its structure, you've got a reasonable idea of its biology, because all the other polyviruses are spread by aphids in a non-persistent manner, blah, 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 blah. So you've actually got an idea of what its biology is, what vector is likely to be spreading it, and where it's likely to be residing. So by understanding its taxonomy, understanding what family it belongs to, you can infer a lot of information based on and again, that's for a brand new virus. Obviously, for existing viruses, that information is already published. So, we're going to finish. That's fine. So finally, a better definition of a virus is from someone else's textbook. That's okay. A set of one or more nucleic acid templates, so it's made of nucleic acid, one or more bits, normally encased in a one or more coat protein, or coats of protein, could be lipoprotein, and it's capable of organising its own replication. So it is an effective unit that once it gets in there can drive its own replication and parasitise the cell. Within the cell, the virus production is dependent on the host protein synthesis machinery. So it needs to use the host ribosomes, a host transfer RNA, etc. to make proteins. It can't produce it itself. And it replicates by organising itself from chunks of coat protein, bits of nucleic acid, sticking the two together rather than splitting cells. And that just says it's not cellular. Okay, let's have a break.